Hello, BIO178. In this video, I'll give a little bit of background about the data set that we are using for our lab this week, which is tied in with the lecture topic of behavioral ecology, and specifically this lab is focused on using molecular data to identify sex bias dispersal. And I think behavioral ecology and using molecular data to inform those topics is super interesting. I don't actually do any of that own type of research in my own lab, nor do I know anyone else at Sac State that does. So the data set we're working with today is actually from a previous publication, which is shown here. This is actually a publication that was cited in your textbook as well. So this is a publication, the title's here, Female-Based Dispersal in the Monogamous Mammal, Evidence from Field Data and Microsatellite Patterns. This is a publication from Proceedings of the Royal Society B. And this data set is looking at the study organism, this little guy here, or girl, I don't actually know if that's a male or a female, but it is a greater white-toothed shrew. These are found in Europe, actually, so we don't have this specific species here in North America, but I'm pretty sure we have other species of shrews. But what they did in this particular study is get samples from 64 female and 76 male shrews near Lausanne, I don't know if I'm saying that right, Switzerland. And if you're like me and your geography is terrible and you had no idea where that was, here are some maps to orient you. So this is a zoomed in version of where it is in Switzerland. And then if you need some more European context like I did, it's located north of Italy, if that helps. Okay, so what did they do? They got samples from 140 shrews total, and this part's actually kind of sad. They got their DNA samples by cutting off the little toes of these shrews. They took a toe from each shrew. From that tissue sample, they extracted DNA, and then the molecular marker they were using in this study is microsatellite. So remember, a microsatellite is a repeat of certain nucleotides. So this example here, this microsatellite is a repeat of the sequence ATAG, and then the allele data you get from these microsatellites is how many repeats there are of that sequence. That's what varies across individuals, across alleles, is how many repeats you have. So they used 8 different microsatellite loci for this particular study. And this is an example of what that data might look like. So each one of these columns is a different microsatellite, one of those 8 microsatellite loci. And this is what the allele data looks like here. So let's start with this first microsatellite locus on the far left. Each individual, so each row is a different individual. Each individual has a genotype indicated here by 2 different numbers. So here, this individual at this particular microsatellite locus has a genotype 40 and 41. So what this means is that that individual has 2 different alleles at that locus, right? So it's a diploid organism. At the first allele, there are 40 repeats for that particular microsatellite. At the other allele, there are 41 repeats. So that's the way this genotype information is read. It's the number of repeats for that particular microsatellite. Some individuals, for whatever reason, weren't able to amplify at that particular locus. In that case, you'll see an NA listed there. So they did that. They used these 8 different microsatellite loci, obtained genotype data for all 140 individuals, and then with that microsatellite genotype data, what they did is calculate assignment indices, or AIC values. The C means that it's corrected, so that's a certain thing that's done in the statistical calculation of this, so we're using corrected AIC values technically. And so what that means is you look overall for the population. So here, these are all the individuals that were collected from population 1. And what you can do is look overall at the genotype frequencies for this population. And then what you can do is for each individual, determine what the chances are that that individual has a genotype that originated from that population, Or if it's a rare genotype, what are the chances that that individual's genotype likely came from somewhere else? So if you look at this first locus, for instance, the first microsatellite column, we can see that allele 40 is relatively common in this population. Allele 41 is common, 59's there a couple times, but allele 39, for instance, this is the only individual that has that allele at that particular locus. So what you can do is take into account all 8 of those loci and see overall for an individual's genotype at those 8 different microsatellite loci, is it likely that individual came from the population it was sampled from, or is it likely it came from another population from somewhere else? And specifically, a negative AIC value indicates that individual came from somewhere else. So when we're thinking about this within the context of sex bias dispersal, what we're doing is once we've calculated those AIC values for each individual, we're separating it out into males and females. And so this is an example image from your textbook where you can look at the AIC values, the number of individuals, and again, remember a negative AIC value indicates that individual likely came from somewhere else. It's an immigrant. And so here in this example, we see females in the darker bar, the black bar, they have more negative AIC values. So in this case, we would conclude that females are the ones that are moving in between populations, and we would conclude female bias dispersal in this case. So this is an example of overall what they did in this study, how they obtained the data. So what we're going to do is have you use that genotype data to then do some calculations in R to determine whether or not sex bias dispersal is occurring.